Proteins do a lot of really cool things, and different cool proteins do different cool things. And the way that they can do that is because they have unique structures that are uniquely suited to those needs. That structure comes in many levels, many layers. We're talking primary, secondary, tertiary, and quaternary. Today, we're going to focus on the tertiary and the quaternary. So, those are going to be your more general overall 3D shape of the protein that's coming from interactions that involve the side chains of amino acids, so those unique parts, those R groups, between the same chain to give us our tertiary structure, or between different chains to give us quaternary structure if we have oligomers. So let's look at where this all comes from and why it matters.
We've been talking a lot about primary structure, so our sequence of amino acids, and how that influences secondary structure, which is the backbone hydrogen bonding that gives us things like alpha helices and beta strands and beta pleated sheets. Now we're going to talk about how those can actually join up to form things like motifs, which are kind of like the furniture within your rooms, and domains, which are kind of like the rooms themselves. And the way that they're interacting with one another is going to be through interactions of the side chains largely. And when we talk about the side chains getting involved, well, now we're talking about tertiary structure. And so tertiary structure is going to involve those intermolecular forces and those interactions between the side chains and between the side chains and the backbone of regions of a single chain. And then sometimes proteins are made up of multiple chains, in which case we can talk about quaternary structure, which is going to be interactions that happen between the different structures, as well as also the whole overall structure itself if you have multiple chains. That's when we're talking about things like oligomers and multimers and all that good stuff. So let's dive in.
So remember, tertiary structure, this is going to involve our side chain interactions. We can have side chain backbone interactions. So we have side chain, and remember another word for that side chain, we're talking about the R group, we can talk about side chain backbone. We can talk about things like side chain cofactor, which we'll talk about. So something like a small molecule that it's bound to, various things when we're talking about the side chains getting involved. And then quaternary structure, not all proteins have. So we have only some proteins. And basically, this is going to be if you have multiple chains. So remember that when we make a protein, we make them as polypeptides, so these long chains. And then some proteins, they have multiple chains. So you would get things like multimers where you have maybe a dimer or a trimer. We'll talk more about these later, but these can be the same subunit or they can be different subunits. So let's look at what kind of interactions we could get in our tertiary structure. And these are the same sorts of interactions that we could also get with our quaternary structure. Just in the terms of the quaternary structure, the interactions are between different chains, whereas in the case of the tertiary structure, the side chain interact and the side chain backbone interactions, those are all between places in the same chain. And so the interactions that can form, well, those are going to depend on the amino acid makeup. And so we talked about how we have those 20 main ones, and we talked about how they all have different properties. And so we have ones that are nonpolar, some that are polar, some that are plus charged, some that are minus charged, various things like this. And we discussed the types of interactions that they can form, and we'll go over these again. But we also have to think about not just these 20 ones, we also have to think about how they can be modified. So basically, you can get what are called post-translational modifications. So remember that translation is the process of making a protein by piecing together those amino acids through those peptide bonds. And this is done by using the genetic information that's held in the messenger RNA or mRNA. So that's the process of translation. And the amino acids are put in place in their original form. But then what's going to happen is that there are enzymes that can actually modify them. They can do things like phosphorylate them. So add a phosphate group, methylate them, acetylate them. There are various post-translational modifications that are going to add extra types of functional groups, extra ways for these amino acids to interact. And note that I've been saying 20 main ones. Basically, these are the ones that you need to know about. These are the ones that our body is going to use. There's one called selenocysteine, which looks like cysteine, but it has a selenium instead of a sulfur. So we use that very, very sparingly. And then some little bugs have, the bugs like bacteria, archaea and stuff, they have a thing called pyrolysine, which is kind of like a weird looking lysine thing. But basically, our bodies don't use that. our bodies use these 20 main ones and they add them like this, and then they can be post-translationally modified. And the significance of that is that they're going to be able to, say, change the function of a protein, alter its activity, add binding sites. Because if you're changing the properties by, like, say, introducing some big bulky charge thing, well, then we talked about how opposite charges attract and like charges repel. So that could influence the IMFs around it, influence the whole protein shape. Or you could be adding a binding site for something because now you have an ion there, which can then form an interaction. So hopefully you're getting a sense that by changing the different functional groups that are available, by changing the properties of these amino acids, we can therefore change what types of interactions can form. And then finally, we can also add extra ways to make interactions by binding to other things that aren't proteins. This is commonly seen with like enzymes, where we have an enzyme that's like by itself. We call this an apoenzyme. So apo is kind of like empty. And then when it binds to a cofactor, we get like a holoenzyme. And what do I mean by these cofactors? So sometimes they could be metals, vitamins, or they can be other small organic molecules. And so basically proteins don't have to act alone. They can bind to metals, they can bind to vitamins and other small molecules, things that are going to allow them to have additional functionality, additional functional groups that weren't present in those 20 main amino acids. So let's talk a little bit about the types of interactions that are involved in tertiary and quaternary structure formation. Now, these are almost all going to be intermolecular forces, or IMFs, those like non-covalent interactions. So these are going to be mostly non-covalent. There's a single exception that we're going to talk about, and that's going to be our disulfide crosslinks. And we'll talk about them first, get them out of the way. And these are going to only form between cysteines. And the rest of them are going to be non-covalent. I should also mention that when we say IMFs, so IMFs, those would be our intermolecular forces. And so typically when we're talking about intermolecular, what we're talking about is between molecules, right? But in the case of proteins, we're actually going to be talking about between regions of the same molecule, so just different places on the same protein. So they're not technically intermolecular, but we're still going to refer to them as intermolecular interactions, and the same principles hold. And what were those principles that we have? Remember, think back on Coulomb's law. We said if we had greater charge, that would be greater attraction or repulsion, but we're just going to be talking in the case of attraction, we're going to be thinking about opposite charges. What else did we say? We said that if we had a shorter distance, that was going to be a greater attraction. And we said that if we had a lower epsilon, so that was our dielectric. So basically that would be lower polarity of the solvent. That would make it easier for those forces to feel one another, and thus you get the greater attraction and repulsion. And again, we're just going to mainly be thinking about attraction, but the same thing would hold in case of repulsion. So when we're thinking about what would make a stronger intermolecular force, we want a greater charge, shorter distance, and less polar solvent. So maybe being in the inside, the hydrophobic part of a protein, rather than on the outside, the hydrophilic part, the surface, where it's exposed to the water, assuming we're talking about a cytosolic water-soluble protein. And so when we talked about the different types of IMFs that we could have, basically we said that our strongest one was going to be our salt bridge. Remember, that's just another term for an ionic interaction. So that's where we have full plus minus charges. And then we had our permanent dipole-dipole. Where we had that special case was going to be our hydrogen bonds, which were going to be the strongest one of those, and we'll review those again in a minute. And then we have just like induced and transient dipoles. So we were talking about our van der Waals interaction. We had our dispersion forces, our nonpolar interactions, and sometimes they're referred to as hydrophobic interactions. But remember that the real driving force of the hydrophobic effect, it's not that these hydrophobic things are attracted to one another. They don't have attraction to one another. Intrinsically, they're just forced together because the water excludes them. And once they're forced together, well, now they have the chance to have those transient dipoles come into play and then make them attract to one another, just temporarily, fleetingly, weakly. But because you have so many of them clumped together, then you can actually see an effect. And so these are going to be the things that we're going to look for. So the strongest of them is going to be the disulfide crosslinks, because these are actually going to be covalent bonds. And so in order of strength, we would have, this would be the strongest. Then we would have our charge-charge interactions. So we'd have our salt bridges. We'd have our permanent dipoles. And we'd have our induced or transient dipoles. Okay, so now let's go and let's look in these in a little more detail. Starting with those crosslinks. So remember, these are going to be our strongest types of interactions, and they're only going to happen between cysteines. So remember, this is cysteine. It has that thiol group, that sulfhydryl group. So thiol is AKA sulfhydryl. So cysteine can deprotonate to form thiolate, and it can also oxidize to form crosslinks. So basically, two cysteines can join together to form this cysteine. I don't know how you actually pronounce things, but basically, when you think about them losing their H's and coming together, you lose that E in the name. So you go from cysteine to cysteine or whatever. And again, I never really say these out loud, but that's technically what they're called. And we often call these crosslinks. Another name for them is disulfide bridges, and disulfide bonds. Those are all names for this same thing. Now, sometimes this happens on purpose and sometimes it happens on accident. So it can happen if you have oxidative species, reactive oxygen around. It can also happen in a controlled manner to introduce site-specific crosslinks, because remember, these are going to be our strongest interactions. So these are going to be strong. And when we're talking about strength, remember, we're thinking about like a single one. So a single one of these is going to be stronger than a single one of our other types of intermolecular forces, because it's covalent. Cysteine crosslinks can play important roles, not only within the same protein, but also between different proteins. So you might see them kind of help hold dimers together, but we'll see also that like a lot of dimers aren't actually going to be held together through crosslinks, but some of them actually are. And you'll see examples of the ones that are, for say things like antibodies, where the different chains are going to be held together by these cysteine crosslinks. Those antibodies need to be really sturdy as they move around the body, and so they're held together by these crosslinks. So these crosslinks are going to be strong and specific, but we have to make them in a controlled manner. And they're typically not found inside of cells because of the reducing environment. And that reducing environment is there to protect our cells. So now let's talk about the more generic forces, the weaker forces. The IMFs are non-covalent interactions. So remember that when we talked about these IMFs, we could kind of simplify things down by thinking of just opposite charges attracting. In the case of a salt bridge, in the case of an ion-ion interaction, well, here you have full charges. In the case of a permanent dipole, such as hydrogen bonding between water molecules, well, that's going to be like permanent partial charge attraction. Then you could also get temporary dipoles form through induction by having something charged come by and kind of push the electrons away as they get repelled by nearby charge or attracted to nearby charge. And then we can have those instantaneous dipole-induced dipole. Basically, because those electrons are all whizzing around randomly, they happen to be in one place more than the other. You get this instantaneous dipole that then induces a dipole in the thing next to it. And if you have a lot of these things clustered together, because the hydrophobic effect due to the entropy gained by freeing waters from solvating things pushes all those hydrophobic things together, and then all those hydrophobic things together have these transient dipole formed, you get these kind of chain reactions that are individually very, very weak, but collectively strong. And so those are the types of IMFs that we're going to be thinking about now. But in terms of which one's the strongest, those would be our ionic ones, right? Because those have full charges. And when we talk about these salt bridges, these ionic bonds, so remember a salt bridge because it's a plus and a minus, and a salt is a cation and an anion that neutralize one another. And so that's what we have here. We have one of our basic amino acids. And then we have one of our acidic amino acids. And remember, when we talk about basic or acidic, that's because we're referring to what it is in its neutral state. So in the case of our acidic amino acids, we have aspartic acid, which in its neutral state is aspartic acid, but we typically find it as after it has already acted as an acid, gave up the protein, and we see it in this aspartate state, its conjugate base. In the case of glutamic acid, we typically see it as glutamate in its conjugate base form, but we name it as acidic because we're talking about, does it act as an acid or a base in its neutral form? In this case, it acts as an acid in its neutral form. What about our basic ones? Well, here we have lysine, arginine, and histidine. In their neutral form, they act as bases. They take a proton and then they become positively charged. So even though we find lysine and arginine and a fair amount of histidine in their protonated form, most of the time, that's going to actually be the conjugate acid form. And so we're almost always going to find lysine and arginine in their conjugate acid form, histidine sometimes, and we classify them all as basic. And when we have one of them interact with one of the other type, that's when we get a salt bridge or an ionic bond. Now, because there's only those few options for these charged ones, unless we're talking about something that's been phosphorylated, well, here there's going to be greater specificity. So not as specific as the cysteine, as disulfide, because there's more options and because they're more common than the sulfur, but more specific than other IMFs. Because there's going to be those fewer options and they're going to be individually stronger. And remember, they're going to be even stronger in like the hydrophobic center of a protein where the dielectric constant is lower. There's less kind of stuff distracting them. They can feel one another better. And so that would be these forces. And remember also that those forces are going to be able to act over a slightly larger distance because you start with a higher charge. So that all goes back to our Coulomb's law. What other types of interactions can we talk about? So we've talked about those disulfide bridges. And remember, those just formed with cysteine. So we had cysteine, so we could have disulfide, our cross-links. Remember that those were often going to be in secreted proteins because our cells keep it reducing environments. We then talked about how we had the basic and acidic. So remember, these would be our acidic, and these would be our basic, and these would form our salt bridges, our ionic bonds. Okay. Well, what would be next? Well, now we want a permanent dipole, right? So we want something that's polar or uncharged. And so these we can form our dipole-dipole. And often this is including H bonding. And remember that our hydrogen bonding was going to be the strongest of our dipole-dipole because they're shorter, because you have that partial covalent bond character. And remember, for a hydrogen bond, we need to have a donor, which is a hydrogen bound to something electronegative, such as an oxygen or a nitrogen. And then for an acceptor, we need to have a lone pair on something electronegative, like an oxygen or a nitrogen. And so if we have a hydroxyl group, if we have a carbonyl group, if we have an amine group, all of these have opportunities to act as hydrogen bond donors or acceptors. And therefore, we can form hydrogen bonds with these amino acids and between these amino acids. We can also form hydrogen bonds between the amino acids and the peptide backbone, in addition to those secondary structure backbone-backbone interactions we saw. Okay, finally, what about the rest of them? Our nonpolar uncharged, these can basically form these can form like dispersion forces. So those just like transient dipoles. And other nonpolar interactions. So other types of VD wall. So basically, these can only do those like transient induced dipole or instantaneous dipole, all those things that are individually weak. Collectively strong. And these are going to be our least specific. So we talked about, we only had a few cysteines, and they had to form under these like certain environments with the help of enzymes, often making things happen the right way. And so we're only going to get those very specific cross-links. But then we said, okay, well, the salt bridges, those were still going to be more specific because we only had a few options that had full charges. And so those are going to be more specific, but not as specific because we still have those few options. When we talk about our polar ones, well, there's a lot of those. And so not only can those polar uncharged ones form those polar interactions, but so could the other ones that were actually charged. Those can also form these. And you can also get them interacting with the backbone. So basically, those are going to be less specific. But the least specific is going to be the dispersion forces, other van der Waals forces, because all of them can do that. And so these are going to be not only the weakest, but the least specific. But remember that the weakest refers to in isolation. And so this is why we can get those big effects when we push them all together. And the big effects come not because they're strong, not because they're attracted to one another, but because of the circumstances. They're all kind of like just pushed together. Remember that the driving force of the hydrophobic effect that actually pushes them together isn't a desire to bind to one another. It's the gain of entropy that comes from releasing water. A special type of those like quote unquote hydrophobic interactions or nonpolar interactions that you'll see is pi stacking. We have pi stacking. More basically what happens is that in a pi bond, in one of these, when you have like an aromatic ring, the electrons are kind of concentrated above and below the ring. And so what can happen is that they can kind of sync up their electron clouds when they're stacked on top of each other. So if you think about there being another one in between, they can kind of like sync up their rings, and then you can get this pi stacking interaction where these are going to kind of attract to one another based on those like dipole things. And so when we talk about the aromatic, this we can also think about pi stacking, which would be one of our like hydrophobic interactions, one of our nonpolar interactions, but it's going to be above and below the plane of these aromatic rings. So that's going to be a special type of one form of those interactions that we'll see. And we can also see that they can make these interactions, say, with the bases on nucleic acids. So we see that we have a lot of different options, and we're going to have a lot of different protein structures. But thankfully, we can kind of simplify things because we can classify them in various ways, and we see the same sorts of repeating patterns throughout them. There's two kind of like main types of protein structures. So we have the fibrous, and then we have the globular. And so on this page are some examples from your book of the fibrous type. So we can talk about keratin. We can talk about silk and collagen. So we looked at collagen a bit when we talked about the secondary structure and how it had that weird left-handed triple helix as opposed to our normal right-handed alpha helix. This one had 3 of these strands that have these weird hydroxyprolines. So there's one of those post-translational modifications coming in. So that would be an example of a post-translational modification. Sometimes we call this a PTM. And remember, that's going to give it special properties. And in the case of this collagen triple helix, it's going to help make this structure. And it's why if you don't have enough vitamin C, you can't make those post-translational modifications, and you make your collagens all weak. And so then you get scurvy. And so, yeah, so your book goes into a little bit of that if you're interested. But basically, this is an example of a fibrous protein. You basically have this repeating units that kind of join together and they make some sort of structural protein. The other type that we're going to be talking about, which we're going to be mainly focused on, are going to be these globular. This is going to include things like enzymes and other proteins that have more like functional roles rather than just structural roles. And there's going to be a lot of different ones, a lot of ones. But thankfully, just like we had those core secondary structures, we're going to see that we have these same sort of structural features that we're going to see in a lot of different proteins, often because they're related evolutionarily or at least functionally. So remember that we have those core secondary structure units. And we talk about tertiary structure. Well, now we're just building upon that secondary structure, right? So we're going to have to incorporate those secondary structures into a larger structure, such as center of picture here, we have this beta sheet in front of alpha helices. And so this would be an example of some sort of tertiary structure because we're involving side chain interactions. We're involving interactions between these different secondary structure elements and putting them together to make a larger fold. So we have those basic secondary structures that can kind of join together to form what we call supersecondary motifs. The word supersecondary is because it's like above the secondary, but it's not like it's hierarchically like you go from secondary to supersecondary to tertiary. No, it's part of the tertiary structure. Even though we call it supersecondary, it's really a form of tertiary structure. So there's a bunch of different types of them. There are things like zinc fingers and beta turns and helix turn helix. From that, you could probably think of, okay, we have a helix, then we have a turn, and we have another helix. So we're putting together these secondary structural units to get these motifs. These motifs may or may not have a known function. For example, zinc fingers, they typically bind DNA. So here's an example of a zinc finger. You can see that it has an alpha helix, it has a beta strand, and it also has a coordinated zinc molecule. So that's one of the places where you can see that not only are proteins getting involved, but actually the other molecules that they're kind of bound to. And this is going to give it this characteristic shape that's going to allow it to bind to DNA. We'll talk more in a second about this word domain, but motifs can be located inside of a larger domain. So the way I like to kind of think about it is like protein motifs are a bit like furniture in a room. So it might be a chair, or it might be a desk, or it might be a table. These may or may not have a known function, but they're often evolutionarily conserved and shared by many proteins. So basically, I might have a chair in my house, and you might have a chair in your house, and the guy down in the street might have a chair in his house. So basically, motifs can be found in a lot of different places because they're functionally useful and because they're just kind of like smaller structural units that are made up of those core secondary structures. And because we have those core secondary structures that are shared between different proteins, well, you kind of expect that motifs would be as well, right? Here's an example of a beta barrel. Sometimes you'll see these making like channels through membranes where you get these pleated sheets forming this circle. You also get things like a beta alpha beta loop. I want you to know a beta barrel, but I don't need you to like memorize like a beta alpha beta loop. But you should be able to look at this and see, okay, well, we've got a beta strand, which is working with this other beta strand. So we've got a beta sheet, right? And then we've also got an alpha helix. So because of the way that this is actually laid out, we have a beta strand, we have an alpha helix, and then we have another beta strand. And so here you can see that although like sometimes people talk about secondary structure as being like between like the adjacent regions on primary sequence wise, this could actually be pretty far away in primary sequence if you had something longer in between. But typically with a motif, we're talking about something shorter, something like this. And then these motifs can actually join together to give you a larger motif. So those are our motifs. And now let's talk a little bit about domains. If motifs were like our furniture, domains are like our rooms. So sometimes these can be distinct rooms, like if you're in an apartment that has actual doors between your rooms. So think of wall separated rooms. But they can also be places in the house where you do specific things. So these can be functional domains, which may or may not be actually separated. So in my old apartment, when I had an efficiency apartment, and it was like one tiny little room where the bedroom was the same as the kitchen, which was the same as the living room, and it was all just one tiny little room. I would do different things in the different parts of the room. So I would sleep in my bed and I would make food, quote unquote, kitchen, which was just like a counter with a fridge and a little microwave across the way from my bed. Those would be kind of like different functional domains. They're all still kind of like within that same area, but I do different things in them. And then my bathroom, well, that actually was separated. And so that would be like a structural domain, but it would also have a specific function. So we talk about a protein domains, we can either talk about them as kind of distinct structural domains, distinct structural regions. Often we think about them as being able to fold independently and function independently of the rest of the protein, but that's not always true. And sometimes we can talk about just like functional domains. Basically, domain is just a way that we can talk about a region of a protein because we need some sort of way to refer to a region of a protein that we want to talk about. And often these domains are going to be things that are going to be shared throughout many proteins, but they're going to be containing multiple motifs often, and they can be these larger regions. So let me just give you an example. So I'll start with a silly example and then I'll go into an actual example. If you think about a Swiss Army knife, well, different parts of it carry out different tasks. So you might have that cork part that's going to be able to uncork a bottle of wine, and you might have that bottle opener part that's able to open your beer bottle. And you might have that knifey part, which is able to slice open an envelope. So we could talk about those as different domains. So here they're kind of like structural domains. I'm assuming that if we took that cork out, still be able to hold its shape. They're also functional domains because, well, they're doing different things. And so we could refer to, say, your cork domain of your Swiss Army knife. This is true for proteins too. They often have domains that are dedicated to certain tasks, but these tasks are often more difficult problems.
In class, we're going to look at an example of domains in action. And so the protein we're going to look at is called polynucleotide kinase phosphatase, or PNKP. It is the helper to your DNA stitcher. So basically, things like radiation can cause damage to DNA, and your cells have an enzyme called DNA ligase that's able to stitch up those ends and piece back together the DNA. But in order to stitch back together that DNA ligase is picky. It needs to have a 3' OH and a 5' phosphate. But sometimes you get the strands broken in a way that gives you a 3' phosphate and a 5' OH. In order to stitch it together, DNA ligase needs help, and it gets it from this PNKP. It needs to do 3 things, and it separates these up into 3 domains. Its FHA domain, well, what it does is it recognizes the damage. Then the phosphatase domain is going to remove the 3' phosphate, because the phosphatase, what it does is it removes phosphate groups. And then at the 5' end, the kinase is going to do what a kinase does. It's going to phosphorylate. It's going to phosphorylate that 5' OH. And now you have the proper conditions for DNA ligase to do its job. So we're going to look at a paper where they actually go and study this protein. So we were looking at the pretty structure, and what happened was scientists had to actually go and solve or figure out this structure. Then they colored it by the domains. And so we often do this with domains. We color them in different colors, and we use that coloring notation in all sorts of representations. And so here you can see that we have this linear sequence alignment, which is going to show you that these domains are actually pretty conserved in some different proteins from different species, as well as like the structural aspects of them in these various domains. And you see that we have the same color coding scheme. We also often see domains in these kind of like domain architectures and topology maps. So we see the domains are kind of colored as basically like squares or rectangles. We'll get more into domain architectures in a minute. And then a topology map where you're kind of seeing the secondary structural elements. So we're seeing those beta strands, those alpha helices in these different domains. And again, we can do this by color coding. We'll also see how domains can contain motifs. So this is a motif that's within one of those domains, and how these domains are often conserved between related proteins.
Speaking of evolution, I want to introduce this term homolog. Basically, a homolog is a gene or a protein that looks and or acts similar because they share an evolutionary origin. Don't confuse them with analog. So an analog is something that looks similar or it acts similar, but it doesn't have a common evolutionary origin. So it can arise because of convergent evolution. So different evolutionary paths, but they stumble upon similar useful things. So they look alike, but they're not actually evolutionarily related, whereas homologs are.
So this is just a heads up of what we're going to be looking at in class, as well as these domain architecture maps. These are really great for showing you how these domains can be linked together in different ways. and different combinations. So we can have the phosphatase and the kinase. So this is what we were looking at before. We've got the phosphatase, the kinase, the FHA, but we can also have the kinase, the FHA without the phosphatase. We can have the kinase, the FHA with other things, just the FHA alone, all these different things. But evolution can kind of mix and match these different domains because we have those sorts of structurally distinct units often. And because they have these really cool functions, those functions can be hooked up in different ways to do different things. And so we're going to look at how we read these domain architecture maps. And so basically, you'll see these domains that they kind of highlight, but then you also see these other regions. Now, sometimes they just don't know what those regions do. Sometimes those regions might have a structure and things, but often these are going to be IDRs. There's going to be those intrinsically disordered regions. And again, these regions might take a shape if they bind to something, and they might have really important roles in, say, like scaffolding and binding to other proteins and bringing them together, but they're not going to be like conserved typically, because there's a lot of different ways you can make a disordered thing, but fewer ways that you can actually make an ordered thing. And then again, we see that the domains are often color-coded. Because these domains are often isolated unit type things, we can often study them in isolation, better understand how they work, maybe even alter them in ways that are going to make them more useful. And so we'll talk more about this stuff in the class, but that is just what to look forward to. And so remember, with tertiary structure, we're talking with a single protein chain. But now let's go ahead and let's think about quaternary structure. And so remember, not all proteins will have quaternary structure, but some of them do. And so this involves when we have multiple protein chains that are acting together. And so if we basically have a single chain, it'll have no quaternary structure. So it acts as a monomer. So mono, one, monomer, one chain. But then we get oligomers and multipers. So when we see the word oligo, this is one of the ones on the core word roots terms I want you to know. Oligo, few, and multi, many. So if you have an oligomer that has a few subunits, a multimer, you have many. Typically, we often just like refer to if we have an oligomer. We can also have pentamer, hexamer, all these different terms, depending on how many subunits we have. Then we can further classify multimers by whether the chains are the same or whether they're different. So if they're the same, then we call them homomeric. So again, going back to why it's important to know those roots. So homo is same, and we could have heteromeric, which would be different. So at least two of the chains are going to be different from one another. So we can talk about a homodimer versus a heterodimer, or a homotrimer versus a heterotrimer. If they're all the same, we can call like the repeating unit. Each chain would be a protomer. And when they join together, then that's when you get this multimer. And so those are different terms that you'll see used to describe the quaternary structure. Again, sometimes you might see disulfide bridges between the chains, but often it's just going to be non-covalent interactions. We'll see that this is going to come into play if we talk about how we might be able to separate these proteins with different experimental methods, and what would happen if you did or you didn't have a reducing agent, because the reducing agent would break up the disulfide bridges, but if you didn't have a reducing agent, then those chains would stay together. We also get to hemoglobin, which it doesn't have those disulfide bridges in between them, but it's got some really, really cool features, and we're going to talk about hemoglobin. But basically, it's a heterotetramer, so it has different subunits, but it has two copies of a beta subunit and two copies of an alpha subunit, at least for the adult version. It's going to bind to iron, and that will help bind to oxygen and carry oxygen throughout the bloodstream. So I hope you're starting to see how the structure of a protein comes from the influence of the amino acids. The amino acids dictate the folding of the backbone. The backbone-backbone interactions give you your secondary structure. The backbone side chain and the side chain interactions give you your tertiary structure. And then sometimes you have multiple chains, like subunits that come together, and then the interactions between those will give you your quaternary structure. All of those give you a unique structure of a protein that is uniquely suited to carry out specific functions. Pretty cool, right?